Hey everyone, as we're waiting for people to come in over the next minute or two, I just want to say hi to you from our group and also introduce you to Dr. Nathan Bryan. So, Gina, tell us a little bit about Nathan's background from one Texan to another.
Well, we're honored and so pleased to have Dr. Bryan with us today. Thank you so much, Nathan, for joining us. I just want to read a little bit about Dr. Bryan. He and I first met, I think it was around 2012, maybe somewhere around in that area, that time frame, when I was working for a group of cardiologists in Dallas. And three or four of the cardiologists were really interested in taking a deeper dive into cardiovascular disease prevention. And so they said, hey, we're going to meet Nathan Bryan and we're going to use this new product to try to boost nitric oxide and see what it does to our patients' blood pressure, because we had read your research looking at brachial artery dilatation, et cetera. So Nathan has a long and very prestigious history. Let me just briefly tell you about Dr. Bryan, who's we're honored to have. Dr. Bryan earned his undergraduate Bachelor of Science degree in biochemistry from the University of Texas at Austin, and his doctoral degree from Louisiana State University of Medicine in Shreveport. He was the recipient of the Dean's Award for Excellence in Research and pursued his postdoctoral training as a Kirstein Fellow at Boston University School of Medicine and the Whitaker Cardiovascular Institute. So after his 2-year postdoctoral fellowship in 2006, Dr. Bryan was recruited to join the faculty at the University of Texas Health Science Center at Houston by Dr. Fred Murad, MD, 1998 Nobel laureate in medicine and physiology. And Dr. Bryan has been involved in the nitric oxide research for the past 20 years and has made many seminal discoveries in his field. That's an understatement. His many seminal discoveries have resulted in dozens of issued U.S. and international patents, and the product technology resulting from his discoveries and innovations has improved patient care worldwide. So Dr. Bryan is a successful entrepreneur and founder of Human N Inc., Newman Nitric Oxide LLC, Nitric Oxide Innovations, and Bryan Nitroceuticals. His product technology is responsible for hundreds of millions of product sales worldwide, and most recently, Dr. Bryan serves as founder and CEO of Nitric Oxide Innovations, a privately held clinical stage biopharmaceutical company that is actively engaged in the discovery and development of nitric oxide-based therapy. And their lead drug candidate, Noviricid, is currently in phase 3 clinical trials for the treatment of COVID-19 in African Americans and Hispanics. We hope to hear more about that. That's very exciting. Dr. Bryan is an international leader in molecular medicine and nitric oxide biochemistry. So again, Dr. Bryan, thank you so much for being here tonight. And as we were talking earlier, we'd like for you to, as best you can in this short amount of time that we have together, give us all an introduction to nitric oxide for those of us who aren't familiar with this miraculous molecule, if you will, and the advantages of using nitric oxide and using all the therapies to boost nitric oxide, and the role that it can play in not only cardiovascular health, but many other areas of health as well.
Well, thank you, Gina, and Whit, it's great to be here with us. Look, it's my honor to be in front of dentists who I think are probably the most important healthcare practitioners in society today. So thanks for the introduction. Thanks for the opportunity to join you. So yeah, as you mentioned, I've been in nitric oxide for many, many years, and I've been very fortunate to stand upon the shoulders of people who've made really great impacts in nitric oxide science and discoveries. And I like to think that we're continuing that research and kind of advancing the field. But for those that don't know, nitric oxide is a gas. It's a gas that's naturally produced in the human body. It has a half-life of less than a second, and so when it's produced, it initiates a number of cell signaling events that are involved in, vasodilation, the regulation of blood flow and oxygen delivery. It's really important in our immune system. We've realized over the past 3 years that, and we recognized this in March of 2020 at the onset of the COVID pandemic, that the people that were getting sick and dying from COVID were the people who couldn't make nitric oxide. And those were people that, primarily the elderly, 50 to 85, African Americans with previous heart attack, diabetes, obesity, smoking, underlying kidney disease, pulmonary disease. And so we actually went to the FDA and I think in June of 2020 and got an investigational drug application to study our nitric oxide drug for, highly susceptible at-risk patients for COVID. And we'll cover that a little bit later. But the reason I bring that up now is it's critically important in immune function, but it's also critically important in neurotransmission. It's a neurotransmitter in the central nervous system. It's critically involved in pain sensitization and nociception. And so the whole point of this is when you lose the ability to make nitric oxide, there are a lot of clinical symptoms that manifest. So number one, you get high blood pressure, typically is the first sign and symptom. Number two, you get erectile dysfunction in both men and women. And what that tells us is if you lost the ability to regulate blood flow upon demand. And so ED is a critical symptom, but it's not just loss of regulation of blood flow to the sex organs, it's loss of regulation of blood flow to the brain. So you get mild cognitive disorders, people develop vascular dementia if not corrected, they develop Alzheimer's, which today is probably the most feared disease around the world because of the burden it puts on family members. So understanding all that about nitric oxide in order to develop safe and effective nitric oxide therapies or product technology, you have to answer the question of how does the human body produce nitric oxide? What goes wrong in people that can't make it? And then once you understand that, now you can start to develop safe and effective, innovative product technology that can impact human health and disease amongst a number of indications. Whit, you're muted. I don't know if you're trying to talk.
I am. So tell us the background, Nathan, when we hear about the three pharmacologists, one of whom you worked with back in '98, winning the Nobel Prize for the discovery of the role of this gas in the human body. What was it exactly that they were explaining to us at that point?
Well, it's besides the discovery of the structure of DNA, it's the quickest awarding of the Nobel Prize from the time of discovery. So this really told us that it was a fundamental discovery and really changed the scope of how we thought about cardiovascular disease and cell signaling. And I think looking back now, as when you can look back of where we are today, you look, it's really three important discoveries in their own right, but the paramount discovery wasn't obvious until you put these three independent discoveries together. So it started with Fred Murad in 1977, who discovered that nitroglycerin, organic nitrates, and these drugs have been used for probably 150 years prior to the 1970s because they relieve the symptoms of acute angina or ischemic heart disease. So physicians used these drugs and prescribed these drugs because they worked, but they didn't know the mechanism or didn't know how they worked. And so it was Fred's work who discovered that these drugs like nitroglycerin are metabolized into nitric oxide. They activate soluble guanylate cyclase. They activate cyclic GMP and then lead to the calcium-dependent relaxation of smooth muscle. So that dilated the coronary arteries, which allowed for the blood flow, and you eliminated the ischemic pain known as angina. So I think in its own right, that's an important discovery, but it's not life-changing, right? You're discovering a mechanism of a drug that's been used for 150 years. And then in 1980, Bob Furchgott, which I think is probably the most profound discovery in vascular biology, he's the guy that discovered that our endothelial cells produced a substance that causes blood vessels to relax. So this is the first discovery of an endogenous vasodilator being produced to regulate blood flow and oxygen delivery. And he called this, in his 1980 Nature paper, he termed this endothelium-derived relaxing factor, or EDRF. So then I think the scientific community at the time realized that was really a very important observation. So then the race was on. What is EDRF? What is this molecule that's produced by the endothelial cells that regulates blood flow, regulates blood pressure? And then about the same time, Salvador Mancada and Lou Ignarro discovered that this EDRF is nitric oxide. And through, like most things in science, they've become political, even back in the 1980s and 90s. Lou Ignarro was awarded the Nobel Prize because apparently he was the first to present this at a scientific meeting in 1987. But Salvador Mancada's paper was published prior to Lou Ignaro's paper in the scientific literature. But EDRF was discovered to be nitric oxide. And so I think the Nobel Prize was awarded because the signaling aspects of a gas was a completely new phenomenon in cell signaling. So now you've got this gas that's produced, it's gone in less than a second. So it just penetrates all cellular membrane. So the question was, how do you determine the specificity and the selectivity of where nitric oxide goes, what it becomes, what it activates? So it was a completely new cell signaling mechanism that had been discovered by these 3 US scientists. So, they made a profound discovery in kind of bringing the pieces of the puzzle together and on how nitric oxide, whether you give an exogenous NO donor or whether you stimulate endogenous NO production, that leads to a number of cell signaling transduction pathways that control most, if not all, biological functions.
Fascinating. One of the ways that we have begun learning about this is through the understanding about nasal versus mouth breathing. And so we'd love to hear more about that from you. We've talked about in our early meetings in this society about, the importance of nasal breathing because of the release of nitric oxide from the paranasal sinuses and how that dilates not only the nasal passages, but all the way through the respiratory passages and in the circulatory system as well. So talk to us from a scientific perspective, your viewpoint on that aspect of nitric oxide in the body.
Well, it's a very important, in fact, it's a fundamental physiological response to what the body is designed to do. So once we understand how the human body makes nitric oxide, now you can start to interrogate which systems go wrong, right? So there's an enzyme in the lining of the blood vessels. That was the enzyme discovered by Bob Furchgott. It's called endothelial nitric oxide synthase. As it turns out, that same enzyme is expressed in epithelial cells. So the largest concentration of the NOS enzyme is found in the nasal epithelium. So similar to the mechanoreceptors on endothelial cells, so when you see an increase in shear stress that's initiated by exercise, that turns that enzyme on. And that's why exercise is medicine in healthy people, because it stimulates nitric oxide production. So as long as that enzyme is coupled and functional, when you do nasal breathing, you get the mechanoreceptors on those epithelial cells that turn on the nitric oxide synthase enzyme. And then that produces nitric oxide. You deliver that nitric oxide to the bronchial airways. It dilates the smooth muscles of the bronchioles, and it leads to dilation of the bronchioles, and you improve oxygen delivery. And that's why deep breathing has been shown to lower blood pressure. The problem is when you have endothelial dysfunction, you have epithelial dysfunction. So the same conditions that cause uncoupling of the NOS enzyme in the endothelial cells cause uncoupling of the nitric oxide synthase enzyme in epithelial cells. And we demonstrated this through exhaled nitric oxide in young people versus older people. So just as you lose production of nitric oxide in the endothelial cells with age, we call this endothelial dysfunction. So the older you get, the less nitric oxide you make in the endothelium. The older you get, the less nitric oxide you make from the epithelium and through nasal breathing. So the key to all this is recoupling the NOS enzyme so that now when you breathe through the nasal sinuses, you can actually generate nitric oxide. But the other problem, kind of the other side of that, is when you do mouth breathing, a lot of these nitrate reducing bacteria that are found in the oral cavity are facultative anaerobes. And so when you do mouth breathing, you're generating a lot of oxygen and you're completely disrupting the microbiome in the oral cavity. So not only are you bypassing the nitric oxide pathway through the nasopharynx, but you're disrupting the nitric oxide production pathway in the oral microbiome. So you've kind of got a dual problem there that exacerbates the underlying condition.
Fascinating. And you're leading right into a second component. So we're trying to understand the different ways in which we can boost nitric oxide throughout our bodies. And we're saying one way would be through nasal breathing and closing the mouth. But as you're talking about these facultative bacteria in the mouth, explain to us what we know little about relative to salivary flow and these bacteria and how that helps with the production of nitric oxide in the digestive tract.
Well, the first pathway to be discovered, and this was through, some studies back in the early 80s, where when people started trying to figure out, okay, what is EDRF and how do we generate nitric oxide, it was recognized that, arginine is an essential cofactor for this pathway. And then once the enzymes were isolated and identified, then this whole reaction mechanism, it's a 5-electron multi-step oxidation reaction that's both energetically and kinetically unfavorable, right? So at least, and this is the time I entered the field in the late 90s, was as a biochemist, we thought, if this pathway is so important, why did nature devise a pathway that's so energetically and kinetically unfavorable? This is a pleiotropic molecule. There has to be another way the human body makes nitric oxide. So that started, and this, probably in the early 2000s, late 90s, this whole initiative on the microbiome project, right? And so most investigators were focused on the gut microbiome. But we started, unlike anybody else in the field, on the oral microbiome, because really disease and digestion and most systemic problems start in the mouth, not only with the foods we eat, but even we knew back then this oral systemic link that people with periodontal disease, gingivitis, oral infections had increased incidence of heart attack, stroke, cardiovascular disease. So there had to be a connection between oral health and systemic health. So that started us on this path of, okay, what bacteria are in the body, in the oral microbiome, that may be responsible for generating nitric oxide or contributing to systemic nitric oxide production. And we look back on the literature, and this literature went back, it certainly predated my time in science, but back in the late 80s, early 90s, a number of investigators, primarily in the UK and at the Karolinska Institute in Stockholm, had recognized that there are oral bacteria that contribute to nitric oxide production. And that identified these as primarily Prevotella, Neisseria, there are probably 4 primary bacteria. So then our focus was on, okay, are these bacteria involved in the regulation of blood pressure? And we published our first paper, I think in the early 2000s, where we created an association between the people that had the highest blood pressure and the least diverse oral microbiome. So the whole point of this was we knew that diet, primarily through green leafy vegetables that contained inorganic nitrate, that when consumed could be activated by the oral bacteria, because 90 minutes after we consume things like kale or arugula or spinach or dark green leafy vegetables, we knew this pathway that nitrate would be taken up in the duodenum, concentrated in our salivary glands. So now for the next 6, 8, 10, 12 hours after we eat a kind of a plant-based diet or a salad for that matter, that our salivary glands would secrete nitrate. And then this nitrate would be activated on by the oral bacteria to then concentrate the saliva in nitrite. So this 2-electron reduction of nitrate by oral bacteria is a true symbiotic relationship, because humans do not have a functional nitrate reductase. So we're 100% dependent upon the oral bacteria. And then now when we swallow our own saliva, which is enriched in nitrite, when that hits the acid environment of the stomach, the pKa of nitrite is 3.4. So at that pH, 50% of the nitrite becomes protonated, and we generate nitric oxide gas in the lumen of the stomach. And then that pathway had already been completely elucidated, because we knew that the nitric oxide formed from swallowing our own saliva would kill things like H. pylori, the ulcer-causing bacteria, would kill E. coli or salmonella or botulism that was maybe found on the leaves of the vegetables. And it enhanced gastric mucosal blood flow, so you can enhance nutrient absorption from the foods you ate. It would prevent ulcerations from chronic NSAID use. So this told us this was again a fundamental physiological process that basically allowed for the production of nitric oxide. But if you didn't get enough nitrate from your diet, or if you had an oral infection and you had oral dysbiosis, or if you used mouthwash or chlorhexidine and you indiscriminately killed the good bacteria along with the bad bacteria, then you disrupted this pathway. We later found that patients who were taking antacids or proton pump inhibitors disrupted this pathway. And to me, that explained why most Americans are sick. Number one, we don't get enough nitrate from our diet. There are 200 million Americans that wake up every morning and use mouthwash. There are 200 million prescriptions written for antacids. That's not counting the over-the-counter purchases. And there are 200 million prescriptions written for antibiotics every year. All of these disrupt nitric oxide production. 2 out of 3 Americans have an unsafe elevation in blood pressure. That's not coincidental. There's a clear association now between the disruption of this essential nitric oxide production pathway and an elevation in blood pressure, increased risk for heart attack and stroke, erectile dysfunction, vascular dementia, Alzheimer's, insulin signaling, and insulin-resistant diabetes. All the major chronic diseases that the world is faced with today can all be explained by disruption, at least partly from a disruption of nitric oxide production.
Fascinating. Well, I'd like to try to combine some things here. As we're talking about the specific oral bacteria that are helpful in that reduction of nitrogen. Tom, of course, has been involved in many, many years of research with analyzing specific bacteria. So is there something happening, or could there be something happening as we can bring all this to the forefront, whereby through dentistry or dental medicine in general, we can begin to evaluate these levels of significant bacteria? Is that something we can do? You've done that in your research, obviously. So is that something that could be made possible to do in clinical settings?
Well, I think so. At least the science suggests that. But just let me express how important it is for basic science to be translated into clinical practice. Because as you guys know, every single human disease has been solved in animals, right? We've solved every mouse disease in the world, from Alzheimer's to heart attack. The problem is none of those translate into human practice or human disease. So this gets lost in translation. So we have to figure out, how do we take the basic understandings of mechanism of action and then translate that into human practice or clinical practice into humans. So I think we're certainly close, and I think we're there, and the science tells us that we can do this. But we have to understand the ecology of the oral microbiome. So I think, from my naive understanding of dental medicine, most dentists are interested in the gingival bacteria that are causing, periodontal disease and gingivitis and caries-causing bacteria. And that's a completely different ecology than what we're interested in on the dorsal part of the tongue. So if you can control kind of the antiseptic effects of the bad guys by maintaining the ecology of the good guys on the dorsal part of the tongue, then I think we can make great strides in selectively killing the bad guys while preserving the ecology of the good guys. But what I can tell you, and most dentists that I've spoken with, and I speak at a lot of dental conferences, when you got a patient with periodontal disease or gingivitis, or just poor oral hygiene or chronic halitosis, the standard of care is chlorhexidine. So you eradicate everything, right? You throw a nuclear bomb in the mouth, and it eradicates the bad guys, it eradicates the good guys, and there are clinical consequences to that. So I think now that we're understanding the ecology of the microbiome, from my understanding and what the research tells us, is that if you maintain a good diversity, then the good guys will keep the bad guys at bay. When you selectively start killing all bacteria, then that creates an environment where the bad guys overpopulate, they take away the good guys, you get dysbiosis, you get periodontal disease, gingivitis, chronic halitosis, and it's maintaining a balance. And I think that's just not in the oral cavity, that's just how human physiology works. So I think now if we can selectively target the bad guys while maintaining, and we have to understand, they're biofilms, right? There's certain levels of bacteria at each stage. And depending upon how you culture these or how you sample these and culture them, most physicians, MDs, for example, think the only thing that's important are the things you can measure, right? I think some of the most important things in human medicine are the things you can't measure. Nitric oxide, for example, it's a gas, it's gone, it's produced, it's gone in less than a second. It's very difficult, if not impossible, to measure nitric oxide production in intact humans. We can measure kind of the results of nitric oxide production and metabolites of nitric oxide production. And I think that goes back to the microbiome and kind of identifying select bacteria that may be present during kind of a pathogenic response, but not necessarily responsible for the pathogen. And I think that's been a problem in the nitric oxide field for 30 years. The best so if you read the literature, you'll find people, and I get these questions every day, go, well, nitric oxide has been shown to be causative for Parkinson's disease and beta amyloid plaque and neurological disease. Well, my answer to that is, when there's a crime, it's, the cops always show up at the crime scene, right? But because the cops are there, does that mean they caused the crime? No, it's just, and that's the problem with nitric oxide. Nitric oxide is always going to be around in any pathological insult because it's our innate immune response to go to the site of infection or go to the site of insult, go to the site of wound or injury, and then kind of corral and be the police to kind of offset that and tape off that level so it doesn't cause systemic disease. So of course nitric oxide is going to be there. And I think it's the same thing that occurred in cardiovascular disease, where you have translocation of bacteria, and you have bacteria that are found in the plaque of people who have suffered from a heart attack or stroke. So sometimes it's a misinterpretation of data. So I never argue with data. The data tell us things, but I think the problem with science and science today, and for many years, it's the misinterpretation of data. And that's very difficult to kind of weave your way through, especially in nitric oxide, because there's, I think, 185,000 scientific papers published on nitric oxide. So the whole point is you can find whatever you're looking for as it relates to nitric oxide.
Tom, would you chime in, please, as a researcher of oral bacteria and, appreciating the amazing work that Nathan's doing? What are your thoughts about this future opportunities that we might have?
Well, first of all, this is still very new to me, Dr. Bryan. My interest has been really in the pathogenic world and identifying them and quantifying them and using PCR and all of that, and we can do that. But as you are stating there, the problem that we have in eliminating the pathogenic species, we don't have a really good option to make sure we're not destroying the nitric oxide producing species. So I think we're kind of in a dilemma there. We have to, in many cases, treat infections to get rid of the disease. And I don't think we have a good choice at this point in time. I think that's what you're saying. Long-term use of antimicrobials presents problems. So what are you seeing in your research relative to short-term use of, let's say, systemic antimicrobials and local antimicrobials? You mentioned chlorhexidine. Sometimes we fight these with local rinses. Sometimes we fight them with site-specific antimicrobials. Sometimes we fight them with systemic. So would you say, I'm just going to ask you the question, if we used a systemic antibiotic, which is very, it's not necessarily targeted like we would like for it to be, but it's targeted as best we can do for these specific pathogens. If we use that short term, what we are seeing, particularly from the Forsyth Institute and the studies relative to the use of antibiotics, even up to 14 days, is that the good bacteria return much faster than the bad ones do, and they kind of push out the bad ones. So what can you tell us a little bit about that? In treating infections, what would you say would be the best case scenario based on what we do today?
Yeah, no, you bring a very good point. And I'm not against, if I'm septic, I want you to give me an antibiotic, right? I'm not against antibiotics.
Right.
I think there's certainly a use for them.
Yes.
But here's what we found in the research is that the microbiome is extremely resilient.
Okay.
And so in a study we published in 2019, we treated kind of normotensive, healthy individuals, 7 days chlorhexidine twice a day. And even that stringent treatment, we don't completely eradicate 100% of the bacteria, right? They're pretty resilient. So maybe 90, 95% of the culturable bacteria we eradicated. But once we stopped, 4 days after we stopped the stringent antimicrobial antiseptic chlorhexidine, the microbiome completely repopulated. And in fact, it completely changed the diversity and the number of good bacteria versus bad bacteria.
Wow.
So I want to make a very clear point here. I'm not against using that.
Yeah.
What I think, because the evidence tells us that you have to, if you've got a severe infection, you have to treat the infection.
Yeah.
And there's always going to be collateral damage.
Yeah.
But what we've learned about this microbiome is they're pretty resilient, and you can completely restructure kind of the ecology to where now you allow the good guys to repopulate and form the biofilms and the good ecology. And it seems to us, at least in our published studies, you can do this within 4 days.
Well, I appreciate that. So what we are doing today, and see if this makes sense, is we are using PCR to qualify and quantify the specific pathogens that we know that are related to this whole story, whether it be oral disease or whether we're trying to connect it to systemic issues there. And our goal is to reduce these pathogens or eliminate, if possible, as Ann Happy would say, to host acceptable levels. And we can measure them, physically or clinically. And also, we can look at inflammatory markers. But we are trying to do this short term. So if we're going to use a systemic antibiotic and a local antimicrobial, our objective is to then do a second test to determine if we've accomplished our goal of reducing the pathogen level, and do we see the clinical signs now improving to health, and then stopping that process and then moving more into, let's say, a homeostatic relationship where we are not using these long term. So we are trying to use them short term. And I think that's what you're saying.
Yeah, absolutely. So, there's a reason we don't take an antibiotic every day for the rest of our lives, right? Because we know the systemic effects of disrupting the microbiome in the gut and throughout the human body.
Yeah.
But an oral antibiotic is dependent upon sufficient blood flow to the active infection.
Yeah.
And in most cases, an active infection is already walled off, right? So you protect that. So what I've found, especially in dental infections and oral infections, an oral antibiotic is not really that effective because you've walled off that active site of infection to blood flow. That's the inflammation and all the hallmarks of infection are that, you go off and you kind of protect that area to keep it from becoming systemic. So I think, there's some clinical utility, but you have to get to and I think that's where nitric oxide comes into play. So if you want to use an oral antibiotic, the beauty of nitric oxide is it dilates not only the resistant vessels to where it normalizes blood pressure, but it opens up the microcirculation and it gets kind of improves blood flow to ischemic or hypoxic regions. And not only is it sites of infection, but it also occurs in cancers and in many things that evade our immune system.
Just one comment on that, if I might, please, and that is that the inappropriate use of antibiotics is what you're saying, I think, relative to this discussion there. We certainly are not going to be throwing antibiotics indiscriminately. What we are doing, though, is very carefully disrupting the biofilm so they become planktonic as opposed to in the biofilm. And we can show, certainly in research and also in our thousands of cases, really tens of thousands of cases, that we can treat these infections very, very effectively. One of the problems, though, is the return of some of these microorganisms relative to once we stop, and helping individuals control this on their own during their maintenance time. That's the struggle. It's not that we can't create an end point of treatment, and that's what we're talking about there, that is very, very positive, but maintaining that is the most difficult part.
Yeah, no, I agree. Anytime you throw a medication, especially an antibiotic, you're disrupting the microbiome, and as a consequence, you're disrupting human physiology because we're dependent upon the microbiome.
Yeah. Yes.
So I think it's important to identify the causative infective agent and
Yes.
eradicate that, which
Right.
I think dentists do a very good job of.
Yeah.
But you also have to begin to recognize the collateral damage and minimize the collateral damage by
Right.
selectively eliminating kind of the bad guys or the infective bacteria. And I think from my perspective, from what I know about dental medicine, is it's a shotgun approach, at least in most dentists.
Yeah, and just to help you there, that's the reason we have a clinical lab today. We were the first to create a clinical lab to actually isolate these. So we know if they are facultative or just simply gram-negative anaerobes specifically, and we're targeting the particular complex that is there short term. This is very new to our profession. And when I say very new to the profession, now we've been doing this for about 20 years, but it's not widely accepted. So what we are trying to do is exactly what you're saying, and that is don't be indiscriminate, but be very selective in what you're doing and be very timely in what you're doing, and use clinical laboratory testing. See, that's the real key there, Dr. Bryan, relative to what we're doing. If you don't know what you're treating, then you are not treating it effectively. If you do know, then you have a much better chance. And then if you follow that up with other clinical laboratory reports to determine if you've accomplished your goal, then you've done a good job. And we're doing a lot better job in doing that by using the lab.
No, I do not disagree at all with that. You got to have a target, and you got to be able to monitor your target and see how effective you are at killing the target.
Appreciate that.
Yep.
Nathan, let's ask a few other things, and some in the chat have asked about molecular iodine. That has gotten a lot of press during COVID and effective as an antimicrobial. What's that doing, if you know, to that balance that we're trying to achieve?
An oral iodine rinse, is that what we're?
Yes.
We don't have any data on the effects of iodine. I mean, obviously, it's an effective antimicrobial. It's used as a surgical scrub before surgeries to eradicate, dermal bacteria.
It's never even above freezing, really.
But iodine is, a part of our innate immune system. Iodine's responsible for our thyroid function. You need iodine for your parietal cells to make hydrochloric acid, and it's, part of our immune function. So we don't have any data, so I don't think I can answer what iodine is doing in the oral cavity in terms of disrupting the microbiome. But I'll tell you personally, I daily take an oral iodine supplement because most Americans, they're deficient in things like magnesium, selenium, and chromium. 95% of Americans are deficient in iodine. So I think it's just good practice to, I supplement with 12.5 milligrams of iodine per day. But that's an oral. I don't think it's affecting, I test my oral microbiome on a daily basis. So I'm very in tune with things that disrupt the oral microbiome. So, a systemic iodine supplement doesn't seem to affect the oral microbiome. An oral rinse, I just don't know. We've never tested that.
Okay. And another question that came up was about hydrogen peroxide. So that's been popular in the mouth. And any exposure to that as far as from a research perspective?
We've never done, and I'll tell you the frustrations we did when I'm retired from full-time academia, but when I was a professor of medicine at UT Medical School and at Baylor College of Medicine, we submit grants to NIH to try to answer important kind of fundamental biological questions. And at no point was the NIH ever interested in trying to answer these questions of what we're doing to disrupt the oral microbiome. So we published a number of papers showing that things like scope, listerine, chlorhexidine obviously do it. But what we don't know, and we don't have the answers to, are things like hydrogen peroxide or essential oils or oil pulling or some of these kind of alternative oral hygienic practices that people use. And so the short answer is we don't know because we don't have any data. But I think with hydrogen peroxide, it's kind of non-discriminately kills bacteria. It selectively kills human cells. So here's what I've learned, and here's what I think. It's not what I know, it's what I think. That if anything you do on a daily basis to kill human cells or bacterial cells is probably not a good idea. If you do this periodically, if you find you have some dysbiosis or halitosis or some oral issue. I think periodically, whether you're doing an ozone rinse or hydrogen peroxide rinse, I think periodically that's may not be a bad idea, but doing that every day, anything that disrupts the oral microbiome every day is probably not a good idea.
Okay. So let's retrace what we've been talking about as far as nitric oxide resources. One is through nasal breathing.
Provided you have a functional coupled nitric oxide synthase enzyme.
Okay. And then secondly, you were talking about through dietary means. So you talked about kale and cruciferous vegetables and dark leafy greens, and just the fact that they're rich in nitrogen that can be then converted. And then you talked about the conversion that happens in the mouth through oral bacteria and that reductase process that goes then through the digestive tract. We haven't talked about a fourth source of boosting nitric oxide, which would be supplementation. So we read and hear about things like beet extract. And so help us, because I'm sure some things are legitimate and some things may not be. And so help us out there, if you would.
Well, it's probably my greatest frustration of somebody who spent 25 years trying to understand the basic biochemistry and physiology of nitric oxide, and then see hundreds of so-called nitric oxide products on the shelves and people are buying. So, the first kind of class of nitric oxide products hit the market in '98 once the Nobel Prize was awarded. And those were products that contain, a lot of L-arginine. So those, and still those products exist today. But the science and really people who understand the enzymology of nitric oxide understand that arginine is a semi-essential amino acid, meaning that we get part of it from our diet, part of it is produced through the urea cycle. And so I'll get into a little bit of complex biochemistry because I think it's important for people to understand the enzymology of nitric oxide. The binding constant, or what we call the Michaelis constant, which is the concentration of arginine needed to saturate 50% of the binding sites to the nitric oxide synthase enzyme, is 5 micromolar. In the sickest of sickest patients, their intracellular and plasma levels of arginine are 100 to 200 micromolar, right? 40 to 100 times higher than what's needed to bind the binding site to convert it to nitric oxide. So there's never a condition where most Americans are deficient in arginine. So it's never made sense to me, why would you supplement arginine to make nitric oxide? And I've used the analogy, it's like putting gas in a car with a blown up engine, right? The problem with your car is not you're out of fuel, your engine's broken to where you can't convert that fuel into energy to propel that car forward. And that's the same thing with arginine. And the problem is, only about 3% of the arginine in the human body is directed through the nitric oxide synthase enzyme. There are other metabolic pathways for it. And if you give high dose arginine to a patient with endothelial dysfunction, they get worse. In fact, in post-infarct patients, it killed more people than the placebo. In patients with peripheral artery disease, it made them worse. Intermittent claudication actually got worse. So L-arginine should not be advised or indicated in anybody with endothelial dysfunction. So that's most people over the age of 40. And it also leads to an expression of arginase. So now you're diverting the arginine that's endogenously produced away from nitric oxide through the urea cycle, through ornithine, through urea exposure or urea production and excretion in the urine. So those products, to me, look, they're a waste of money. It's been a joke in the nitric oxide field for 40 years now. But you mentioned beetroot and beetroot extract and beetroot juice. Those hit the market, I think, about 2012, because there was some published literature coming out of the UK that showed that a lot of athletes were drinking beetroot juice and you could improve their performance, improve oxygen efficiency of ATP production. And in the 2012 Olympic Games in London, it was realized that in fact, the UK Olympic team won more medals than any time in the history of the Olympics in 2012. And they attributed that to their beetroot juice they were drinking. And then there started to become a story around, okay, beetroot contains nitrate that's made into nitric oxide in the human body, and that improves mitochondrial efficiency of oxygen utilization, improves, pushes back the anaerobic threshold, improves performance, less lactic acid buildup, and improved recovery. So then the market was kind of overrun with beetroot products. And today there are dozens, if not hundreds, of beetroot products. And, I have 2 to 3 dozen issued patents from the University of Texas owns when I was employed there. So I have an obligation to test these products to make sure, number one, they're not infringing on any patents, but number two, to figure out, are they actually doing anything as it relates to nitric oxide? And I can tell you, I've tested almost, if not all, beet products on the market, and 98% of those do nothing. The only thing they do is turn your urine and poop red and pink and cause a lot of anxiety. I can't tell you how many calls I've gotten at night from people in the ER thinking they have a gastric bleed, and they end up being okay. And I go, well, have you taken any beets? And they go, oh yeah. Well, that causes a lot of anxiety, but it does nothing to nitric oxide production. So we call those dead beet products because they're really dead beets. They do nothing except cause a lot of anxiety. So when we started trying to integrate and make innovative nitric oxide products through beets, we started at the source. So it starts with the type of seed you buy, the soil conditions you plant those in, the time of harvest, and then the most important thing, once you harvest those beets, you get what's called a high-brix concentrate, or kind of like a product that looks like molasses. It's kind of really dense, viscous, and then you got to dry that and make it into a powder. Well, most companies, to save money, they just put it through a high heat, high pressure drying system. And these nitrate and nitrite, and in fact, a lot of these nutrients are water soluble. So they just come out, right? So the finished product has no nutrient density whatsoever. What we look at is primarily nitrite and nitrate, but other water soluble minerals and vitamins are completely lost too. So there's a lot of companies out there selling a lot of beetroot products that are marketed as nitric oxide that have no nitric oxide activity whatsoever.
Well, then tell us we want to hear more about your work as far as from that standpoint of creating products that do work.
Well, this took probably 15, 20 years to understand. You cannot even begin to contemplate developing nitric oxide product technologies until you understand how the human body makes it. So what we do, and especially in our drug company, most drug companies create synthetic compounds that inhibit endogenous enzymes, right? And that's why there's always side effects of drugs. So if you inhibit the body's ability to make cholesterol by giving an HMGA-CoA reductase inhibitor called a statin to inhibit cholesterol synthesis, there's always side effects, because that's nature's way of providing a source of cholesterol that if you're not getting it from your diet, the body's got to make it. It's essential. So there's always side effects. And this goes for most drugs, right? There's protease inhibitors that are now being introduced there's a COVID therapy. These go back to the 1980s for HIV drugs that are very dangerous. So the whole point is, what we do as a drug company is we don't make inhibitors of enzymes, because in most chronic diseases, and in fact, there's no indication, there's no human chronic disease where overproduction of nitric oxide is a problem. So there's no need to ever inhibit nitric oxide production. In fact, most chronic diseases are caused from insufficient nitric oxide production. So how do you replete and restore nitric oxide production? So number one, you got to figure out how much nitric oxide is produced on a daily basis that humans are normally exposed to, right? Because we want to recapitulate physiology. We want to give the body back what it's normally designed to see, or what it normally produces. And you can't do this until you understand both production pathways. How much nitric oxide are we getting from the diet and kind of a standard dietary patterns? How much nitric oxide is being produced from the conversion of arginine to nitric oxide. We understand that through radio-labeled isotopes from the conversion of arginine to citrulline. And so now we understood how much nitric oxide we need to produce. And nitric oxide is a gas. And I think, early in my career, I consulted for a number of companies, including GlaxoSmithKline, Bayer, AstraZeneca, because most of these companies had nitric oxide drug discovery programs where they were trying to make nitric oxide drugs. And to date, every single major pharmaceutical company has failed at generating a safe and effective nitric oxide drug. And the reason is their approach is wrong, right? So we have to recapitulate the production of a gas that's gone in less than a second, but then activates a number of signaling pathways. And so what we do is just that. We figure out how much nitric oxide we need. How do we deliver nitric oxide systemically to every organ, tissue, and cell in the body that recapitulates normal signaling aspects of that? And we couldn't do this until 2007, when we were the first to report that nitric oxide acts as a hormone. In this paper we published in the Proceedings of the National Academy of Science, and we were the first to demonstrate that nitric oxide produced in one biological compartment had distal effects on every tissue in the body. So then this told us, well, all we have to do is generate nitric oxide in a single biological compartment. So we started in the oral cavity. And so we developed an orally disintegrating tablet that when you put it in your mouth, as this matrix is falling apart over 5 to 6 minutes, it generates about 20 to 30 parts per million NO gas. It's vasoactive. We can see dilation of the carotid arteries within about 12 seconds. In our animal studies, we could protect from injury from heart attack. We could protect the liver from injury from a number of insults. And so that's how we've been able to develop safe and effective therapeutics. And then kind of understanding that people were using beets and beetroot products as a source of nitric oxide, we knew that this started with nitrate, right? These vegetables accumulating nitrate from the soil, that then when consumed, if the patient had the right oral bacteria, they could convert this to nitrite, and then sufficient stomach acid, they can convert this to nitric oxide. But I would never bring a product to market that worked in 10% of the population, right? Or 20% of the population. Because what we're finding is that, 2/3 of the population use mouthwash. 2/3 of the population use antacids or some type of acid blocking. So that tells us, well, that's 400 million people. That's more than the whole population of the US. So if we just gave nitrate, then perhaps 10%, 20%, 30% of the population would get a nitric oxide effect. So then what we did was we pre-converted. So we used a fermentation process that we could pre-convert the nitrate into nitrite, into the beetroot products or into our lozenge, and then basically bypass the need for the oral bacteria. So what that means is if you've got a patient with chronic halitosis or on antibiotics or chlorhexidine, and you give them our nitric oxide product, this product's going to work whether they have the right bacteria or not, because we've pre-converted that for you. We don't depend upon the individual person's microbiome or individual person's stomach acid production. If your body can't make nitric oxide, we do it for you. And that's the beauty of what we do versus anybody else. And there's no other product technology in the market that I'm aware of that does that. So that's kind of the difference that we do. But again, it's not my job to sell people products. My job is to, create education and provide information so that both physicians and dentists and patients can make informed decisions. But that's a big challenge, because all these companies say the same thing, but yet very few products actually deliver on generating nitric oxide.
Can I ask a question?
Of course.
Good. Have you done any research in using your nitric oxide as a treatment entity for these particular pathogens that we're looking at? Have you looked at these pathogens specifically, and then the nitric oxide use that you're using, or seeing a difference there?
Yeah, we have, and many others have. So most pathogens are anaerobes, as you guys are aware.
Right.
And most anaerobes are very sensitive to nitric oxide. In fact, that's how our innate immune system kills off a lot of these. So the mechanism is that if you generate nitric oxide, the nitric oxide being produced binds to the iron sulfur centers of these anaerobes, shuts down their respiration, and selectively kills the bad guys. So we can do that. We've been able to do that with things like botulism, H. pylori. We're looking at some of the oral pathogens, but I don't recall. We've got some data in the works now.
Could we follow? Yeah. Could we follow? Could we follow through with that? I would love to follow through with that at some point in time, because this is what we're searching for, and that is, treatment that doesn't destroy the good ones but destroys the bad ones, and we can measure that clinically, and also we could measure that even systemically, in various ways, whether it be inflammatory markers or CIMTs and so forth.
Well, I'd love to provide you some of our product technology to test that. We've had a number of dentists that do it in their office.
Yeah.
That use the dark field microscopy and the spirochetes. So we know that, we can, we see a vast improvement in that.
Yeah. Well, what we're using is DNA, of course, and we're using PCR. So we're really quantifying. I use phase contrast for many years, but of course we have to be able to know specifically what we're looking at. Many of these are too small to look at phase contrast, even at 400 power. So I would love to follow through with that if you would be willing to do that.
No, we'd love. Look, I've been trying to find a dentist to do this for probably 10 years.
Well, we have a laboratory that does that. So, we can do this very highly specific. So if Whit can keep us in touch, I'd love to work with you on that.
For sure.
Right.
Yeah, we've got a lot of anecdotal evidence, but for me as a basic scientist, you have to be able to reproduce this. Look, an N = 1 experiment is very important.
Good.
But to make it meet the rigors of publication, you've got to be able to reproduce this in a number of different people.
I'm on top of this, and I hope you can help me with this.
Absolutely.
Great. Thank you.
That's a priority.
Great. Thank you.
Hey, Nathan, one of the things that we've learned from you and your work is that over time, as adults, our nitric oxide levels begin to really drop after about 40 years old. And so you've alluded to this, and we want to pursue it with you further. And that is, we need to really boost it up, and I think a lot of us on this call are over 40, but certainly our patients and the overall population. So what would your recommendation be in boosting nitric oxide levels on a daily basis? And I know you don't want to be proprietary, so we're just going to ask you, what do you do? And then
it's probably the most
proprietary.
It's the most common question I get. I just turned 49 in November on Thanksgiving, and but I do routine vascular aging through endothelial function. I have the vascular age of a 32-year-old, and I'm a Scotch draining, meat eating Texan. And I'm on a plane every week for the past 4 years. So I live a fast paced, kind of a high stress life, but I do things routinely to make sure I optimize my nitric oxide production. So I tell people, you only got to do 2 things. Stop doing the things that we know disrupt nitric oxide production, and start doing the things that are clinically proven to promote it. So now if we take those step by step, so what disrupts nitric oxide production? Number 1, if you're using mouthwash on a daily basis, you have to stop. There's clear evidence now that mouthwash causes an increase in blood pressure, which is the number 1 modifiable risk factor for cardiovascular disease, the number 1 killer of men and women worldwide. I was on the doctor show 2 years ago where we revealed that if you use mouthwash and you exercise, you lose the cardioprotective benefits of exercise. So not only is it, so you can do all the right things, eat a plant-based diet or eat a lot of vegetables and exercise, but if you use mouthwash, you completely eliminate the benefits of that. So I think you have to work, and I think this is why what you guys do is so important, because it all starts with oral medicine and the dentist. Number 1, you have to get rid of the oral infections, but you also have to start taking into account the collateral damage you're doing by disrupting the oral microbiome. And then number 2, you have to get rid of fluoride. And this causes a lot of indigestion with dentists because they want to do fluoride rinses, but fluoride is an antiseptic. It kills your thyroid function. It's a neurotoxin. And so you've got to limit your thyroid or your fluoride exposure. Because fluoride, if you guys remember, is a halogen. It's on the same column as iodine. And we have an epidemic of hypothyroidism. So most Americans are deficient in iodine, but they're replete in fluoride because it's in their drinking water, it's in the water they cook in, they bathe in, and it's in their dental rinses or their mouthwash. So you got to limit your fluoride exposure. And then number 3 is you got to stop taking antacids. Antacids completely disrupt nitric oxide production. There was a paper published, I think in 2016 by my good friend John Cook, that showed that people who have been on PPIs for 3 to 5 years had a 40% higher incidence of heart attack and stroke. PPIs completely disrupt nitric oxide production. So those are the 3 big ones. Stop mouthwash, get rid of fluoride, stop using antacids. Now you start doing the things to promote it. So throw in some more green leafy vegetables, salad, spinach, kale, moderate physical exercise, 20, 30 minutes of aerobic exercise a day, 20, 30 minutes of sunlight or an infrared sauna. And then if all else fails, we have product technology that does it for you. So despite the fact that I travel every week, when I landed in New York City a couple of days ago, first thing I do is I go to the gym and I work out an hour. I go to a place who has an infrared sauna because it's cold and gray and rainy here, and I need sunlight. And at home, I sit in an infrared sauna, have a hyperbaric chamber at my house. But then I take our nitric oxide products every day, and I try to, control and maintain my stress level the best I can. But, this is very challenging. It's hard work staying healthy, but I can tell you, it's easier to prevent disease than it is to treat it.
Tell us again the name of your company.
Well, I've got 3 companies. We've created a number of nutritional and dietary supplement products. One's called Bryan Nutraceuticals, where we have products at no2u.com. We offer wholesale pricing to practitioners. We developed a skincare company because we realized a couple of years ago the skin is an organ, and without sufficient blood flow to the skin, that organ fails. So we have a company called Pneuma Nitric Oxide. That's n101.com. We got a topical nitric oxide serum for fine lines and wrinkles and a lot of aging related problems with the skin. But what I'm most proud of our drug company, because we're actually going through the FDA to develop safe and effective FDA approved drugs for a number of chronic diseases. We just finished a study in COVID in African Americans and Hispanics, high risk population for COVID. We've got a drug for ischemic heart disease that doesn't develop tolerance. Nitroglycerin have been very effective for 200 years, but it's limited in its clinical utility because people develop tolerance to it. Our drugs dilate the coronary arteries, don't develop tolerance. We've got an Alzheimer's drug now we're starting on. We've got a topical drug for diabetic ulcers, non-healing ulcers. We're taking 3 to 4-year-old non-healing ulcers. We apply our topical nitric oxide and we heal these non-healing wounds, in a period of 3 to 4 months. And to me, that's really life changing. And it first got me interested in this because my dad's a paraplegic from a car accident in 1984. So dad's 70, he'll be 74 years old this year. And he's been in a wheelchair since he was 34. So he spent more than half his life in a wheelchair. So I've dealt with these, and I've been treating decubitus pressure ulcers since I was 12 years old. And the one thing that I've recognized is when we had these non-healing ulcers, I could heal it with a nitric oxide releasing gauze. And, we took non-healing ulcers and with dad and, saved his life on a number of different occasions. So, there's been no innovations in wound care for 50 years. And I think there's 600 people that die every day in wound care facilities from septicemia and non-healing ulcers. And I think, the power of nitric oxide of killing the active infection in the wound, causing the hyperemia, tissue granulation, and wound healing will transform wound care. And, there's really not a single indication where nitric oxide given at the right dose at the right time in the right patient wouldn't be beneficial. And that company is called Nitric Oxide Innovations. And, I think that's where my effort is today. These nutritional and dietary supplement products and skincare products are important. There's a huge market for them. But, to me, there's nothing more important than going through the clinical trials and not only showing safety of our drug technology, but showing efficacy in a number of poorly managed chronic diseases.
That's very exciting. Another question that came up through the chat was about xylitol. Have you had any work that you've done with that?
You know, I've certainly followed the literature on xylitol and kind of affecting kind of the gingival bacteria and kind of being effective at eradicating the bad guys in terms of oral pathogens. What we don't have data on is what does this do to the nitrate reducing bacteria?
Mm-hmm.
So as you guys know, there's a number of oral bacteria that are involved in a number of biological functions. We're focused on kind of a single kind of activity of multiple species. So we just don't have any data on what it's doing to these bacteria.
Mm-hmm. Mm-hmm. Okay. All right. Gina, do you have any additional questions or thoughts you want to share with Nathan?
I think another interesting, we were sharing the website, both your Nathan Bryan website and the N101 and N02U, but I think an interesting comment is that we need a wound healing nitric oxide mouthwash or a topical for the mouth. Are you looking into that?
We are. You know, I've been playing around with a mouth rinse because I realized the importance of oral rinses and kind of selectively killing the bad guys by maintaining the integrity of the good guys. So, you know, I've been kind of investigating this for more than 2 years. I think we have a formula and a mouth rinse that's going to do just that. Obviously, we've got probably several months worth of testing. I had hoped to launch this in the first quarter of 2023, but we just don't have the data that we need to feel confident that we bring this to market and can, you know, appease, you know, people like Tom and who are going to obviously put it to the test. And so I will never launch a product until I feel 100% confident that what we're doing is intended to what is going to do what it's intended to do without, you know, causing any collateral damage. So we've got a little bit more testing, but I feel certain, you know, probably within this calendar year, we'll feel good about launching a mouth rinse because, you know, there's a number of people that use mouth rinse as part of their daily hygienic practice. They want a clean feeling mouth.
Yep.
But we have to be able to do that by maintaining the ecology of the oral microbiome.
Love that. Boy, that's great.
Mm-hmm. Well, you may have a few investors on this call tonight.
Contact me. You know, we're actively raising money. I've self-funded this for, you know, the first couple of years, but you know, we feel very good about where we are, the safety of our nitric oxide drug and COVID. You know, now the FDA is allowing us to go straight into a phase 3 clinical trial in all of our other drug indications. So those of you that know anything about drug discovery, phase 1 and phase 2 trials typically take, you know, 4 to 7 years and anywhere from $20 to $60 million. So now we bypass that and go straight into phase 3 trials for any of our oral formulations.
If I could ask you again, I mentioned your findings that nitric oxide levels really begin to take a dive after about 40. Why do you think that might be?
Well, that's from the arginine pathway. So that's what we call endothelial dysfunction. So the activity, the uncoupling of that enzyme in the endothelial cells. So all of these studies derive from doing intra-arterial infusions of arginine or some agonist like bradykinin or acetylcholine to stimulate or activate endogenous nitric oxide production. So what that tells us is we lose about 10 to 12% of our endothelial function per decade. So by the time we're 40 or 50 years old, we only have about 50% of our endothelial function compared to where we were when we were earlier. So there are a number of things that contribute to that. A lot of it is diet and lifestyle. You know, some of it is these single nucleotide polymorphisms in certain enzymes or SNPs in the MTHFR or eNOS that render us, you know, nitric oxide deficient. But you know, it's multifactorial, but it doesn't have to be that way. So for instance, you can overcome endothelial dysfunction by compensating for nitric oxide being produced through this oral enterosalivary circuit.
Wow, this conversation is so mind-blowing, Nathan. The work that you've done is revolutionary. We would love to see you win a Nobel Prize, and we'll all come celebrate. So, I mean, the things that we've learned both about nutrition, obviously appreciating nasal breathing, what goes on in the mouth, and the effects of mouth rinses. Fluoride, that's a new discussion for many of us tonight that's kind of astounding, but could certainly make a lot of sense. We're so excited to learn more from you. And I'm going to go ahead and begin to wrap things up because I think we've covered a lot of ground tonight. Thank you for that. And Nathan's been at meetings all day today, and he's in a hotel room in New York City, even though his home base is down in the middle of Texas. So he's away from his family in a warmer confines in New York City than where his wife and children are, with the power out in Texas, having a freeze there. So I know that's kind of something that's on your mind tonight as well. But I just want to let everyone know how excited we are that in our next big event coming up, that I hope every single one of you will be there, is going to be in St. Petersburg, April 20th and 21st. And we're doing our IDM Scholar Society symposium. We're calling it the IDM Revolution, but it's a symposium, and Nathan is coming to be with us. He's going to bring his partner in business, and he's going to be speaking at that meeting along with 15 other experts. And we have, I think, 6 dentists, 5 hygienists, 2 PhDs, 3 physicians that are going to be at this just tremendous meeting. And you'll have a chance to meet Nathan in person there. That is April 20th and 21st in St. Pete. So please go ahead and sign up for that and come join us. This is going to be kind of our first reunion type meeting for our study club, our first live event since we've been meeting. And please bring your colleagues, both dentists, hygienists, nurses, nurse practitioners, physicians. We're going to talk about nitric oxide and peptides and a variety of really exciting subjects that are new to most of us, just as we've been learning tonight. And so, Nathan, thank you for taking time tonight. And I know you must be exhausted, and then a whole nother lecture that you weren't necessarily planning on giving tonight. But I know how passionate you are, and we're just excited to get to know you better and to learn from each other and learn from you on this lifetime of research that you've done. And so, Kim, is there anything else that we need to cover tonight before we wrap up our evening? And for some of you, go have dinner, and for some of us, go to bed.
I think that pretty much covers it. We do always have the study club discount code for our events. So if you all are registering for our events, SC10, like Study Club 10, you can put that in, and that will give you 10% off most of our offerings. So anyone that comes to participate in these groups, we want to make that available to you. And we just appreciate everyone that's joined us. I don't think I have anything else at all.
Okay. If you're planning, and I assume you are, to come join us in April, I would recommend that you get your hotel reservations soon. St. Petersburg and Florida is just busting, and it's a very popular destination year round, but I would get your hotel reservations early, just as a heads up. And you could stay with us, but we already have a whole group of people staying at our house. So I'm afraid we don't have any room. We might have one couch left. But anyway, we're limiting this. We're going to do it at the St. Petersburg Yacht Club right down on the waterfront. It's a gorgeous venue, and they can hold 140. So we have 15 speakers and our partners. So we've got about 120, no, maybe 100, maybe 30 accounted for before we even start. And we've had a number of people register already. So don't wait till the last week, because you won't be able to join us. We really want to make this a big event. So Nathan, thank you so much for taking the time tonight. And I know you need to call your family and see if they're surviving. But thank you for the time tonight. And thank you all for joining us. And we'll be in contact and come join us in April and bring a friend or a colleague or a team member.
Yeah.
Thank you, Tom and Gina,
Yeah.
as always,
Appreciate
for
that.
Yeah. Thank you
helping
all.
us. Y'all have a good evening and be safe. Take care now.
Thank you. Yeah, bye.
Good night.